seed biomass. We took the best plants out of every generation and made the selection procedure for 4 generations. Here we made selection on the basis of survival and on the basis of fitness, when our fitness criteria was seed biomass. So after 4 generations, we analyzed these plants of the 4th generations. So we put control selected plants under control condition and under heat stress condition. We put heat selected plants under control condition and under heat stress conditions. So I'm now going to go to the next section of my presentation, that is a result section. So here in this section I'm going to answer these 2 questions. What is the impact of combination of heat and drought on plants, and how does elevated CO2 alter these responses? So here we can clearly see that control plants and elevated CO2 grown plants, CO2 plants are already bigger, and the impact of heat and drought is very clear here because the plants are smaller, and CO2 plants are bigger under stress. So it is quite interesting that the CO2 is mitigating the impact of stress on plant growth. So it becomes more clear here when we measure rosette biomass, and for example this represents our control. This is non-stressed elevated CO2 plants, and this is the effect of heat and drought in the presence of elevated CO2. So what is happening? So the reduction in the absence in the ambient CO2 is 76%, but in elevated CO2 it becomes only 52%. For example, here this arrow represents when it is unfilled, the impact of stress. When it is filled, the impact of CO2. So we can say that elevated CO2 is mitigating the impact of stress on plant growth. So it was quite interesting, so we wanted to see what mechanisms are changing or what is happening on the molecular machinery of the plants. So we did genome-wide transcriptional analysis by using microarray. We can see with this Venn diagram, it represents the differentially expressed genes in Arabidopsis during the heat and drought exposure and during the heat and drought in the presence of elevated CO2. We can see heat and drought have a massive impact on the transcriptome, and this impact is lessened by the elevated CO2. And we can see from here also the majority of the genes, approximately 2,500, are commonly shared by both the stresses. So the stress impact is higher, but elevated CO2 is doing something, but it is lessening the stress impact. So what is happening on the metabolism? So this graph represents the whole metabolism of Arabidopsis. For example, this represents photosynthesis in the form of light reaction. This represents photorespiration, for example, chlorophyll related genes, and starch sugar metabolism related genes, lipid related genes, and secondary metabolism, amino acids. So this color represents when the gene is downregulated, it is in red color in these blocks. When the gene is upregulated, it is in blue color. For example, here. So what we see here is various metabolic pathways were changing, but photosynthesis, most of the genes of the photosynthesis were downregulated. Along with, there were changes in the sugars and amino acids. So we did the same kind of analysis for the plants in the presence of CO2. So what comes out? So interestingly, the impact of, or you can say the negative impact of CO2, which was in the form of downregulation of photosynthesis genes, was reversed by elevated CO2. So from here we can say that elevated CO2 mitigates the impact of stress on photosynthesis. So we were quite interesting what is happening on photosynthesis lab. We measured photosynthesis, we measured chlorophyll fluorescence, which represents the efficiency of photosystem 2. We can clearly see from here that photosynthesis was downregulated by the stress. The photosynthesis, and in the presence of elevated CO2, the downregulation was lesser. The same also becomes true for chlorophyll fluorescence, which was measured in the form of Fe over Fm. There was downregulation of the Fe over Fm ratio, but the elevated CO2 mitigate this effect. So we wanted to see how what is happening on the level of the oxidative stress. We measured different oxidative stress parameters like cross production. We measured here hydrogen peroxide content. We measured membrane oxidation in the form of myelin dialdehyde, which is the aldehydic production of membrane oxidation. We measured protein oxidation in the form of protein carbonates. So what we can see here is heat and drought stress result in the accumulation of the ROS, or specifically saying accumulation of the hydrogen peroxide. But CO2 lessen this effect. And interestingly and surprisingly too, that the MDA levels were going down during our stress treatment. And we can see that elevated CO2 further decreased the accumulation of the malondialdehyde. But protein oxidation was increased by the stress exposure, and this effect was lessened by the elevated CO2. From here we can say that climatic extreme, or specifically saying the heat and rod treatment, resulted in oxidative stress. We can see from hydrogen peroxide and protein oxidation, but result in protection of the membrane. So we further went further to measure different kinds of molecular antioxidants. Like here, this graph represents the total antioxidant capacity of the plant cell, and this represents the total phenolics, and this represents tocopherol. Tocopherol is a membrane bound antioxidant. So what we can see here is the